The enzyme used in this experiment was peroxidase. Enzymes are catalysts that lower the activation energy of a reaction and therefore speed up a reaction. Enzymes can be reused over and over again. Peroxidase is a large protein with an iron ion acting as a cofactor found on its active site. It is commonly found in turnips, horseradish, and potatoes. The substrate is hydrogen peroxide, and this enzyme rids cells of hydrogen peroxide, which can damage cells if not removed. Peroxidase reacts with hydrogen peroxide to produce water and oxygen.
These test tubes contain a mixture of a pH 5 buffer, H2O2 extract, or guaiacol. The blank has 6 milliliters of buffer and 1 milliliter of extract with 1 milliliter of guaiacol. Test tubes 2, 3, 4, 5, and 6, 7 will be mixed together in a cuvette to be put in the spectrophotometer.
The test tube contents were mixed together in the cuvette with test tubes 2 and 3 mixed in one cuvette, 4 and 5 in another, and 6 and 7 in the last.
This machine is called spectrophotometer, and it is used to measure how much light passes through the cuvette. The cuvettes have a solution containing guaiacol and enzyme stripe. As guaiacol reacts with enzyme, the reaction darkens. As the solution darkens, the absorbance rises. By measuring the absorbance over time, we can measure the rate at which the enzyme reacts with the guaiacol.
Mixing was done within 10 seconds, and the cuvette was immediately placed in a spectrophotometer that was set to 500 nanometers. The absorbance was recorded every 20 seconds, and this was repeated for the other two cuvettes. This was the method used for all of the cuvettes. 2 test tubes were mixed in one cuvette and then placed in the spectrophotometer, and absorbance was recorded every 20 seconds.
Every single mixing?
Yes. I'll start. You can start when the 4 goes in.
The absorbance for the test tube 4 and 5 after 1 minute and 3 seconds. The absorbance is 1.999.
When test tube 4 and 5 were mixed, after 120 seconds, the enzyme reacted with the guaiacol to produce this orange-brown color. It's dark.
Okay, let's do... To determine the optimal pH for peroxidase, we have different pHs tested for a rate of peroxidase activity: pH 3, 5, 7, and 9.
tubes 4 and 5 were mixed, they reacted quickly, changing the color to a light orange. Okay, yeah, we're done. This graph shows time versus enzyme concentration. Test tubes 2 and 3, the blue line, is the most linear because it is the optimal concentration of enzyme. This concentration, 0.5, was then used for the experiments on temperature and pH. This graph shows how temperature affects the rate of reaction of the enzyme and guaiacol. At a temperature of 100 degrees, the enzyme and guaiacol rate of reaction is relatively slow. When you increase the temperature to 48 degrees, the rate of reaction also increases. At a temperature of 4 degrees, the rate of reaction is relatively fast as well. When you increase it to 32 degrees, the rate of reaction for the enzyme and guaiacol has increased even more. Finally, at a temperature of 23 degrees, the rate of reaction is extraordinarily fast and reaches 1.99 absorbance at a very quick rate and stays there. The optimal temperature is 23 degrees. At 23 degrees, the rate of reaction for the enzyme, peroxidase, and guaiacol is the fastest. All other temperatures are not as fast or as optimal as the 23 degrees. And this line in this graph is the slope of the previous graph.
As the pH level gets higher, the enzyme's activity also gets higher. This graph shows that at a pH level of 9, the enzymes were working very well, compared to a pH of 7, which shows that there was only a little activity. At pH levels of 5 and 3, the enzyme was suitable to work. The pH that is optimal is a pH of 9. When taking the slope at each pH level, it showed that at a pH of 7, the enzyme did not work as well as pH levels of 9, 5, and 3. On this graph, the rate of reaction at each level is shown.